Okay, moving on. We have this example here that is very similar to the example that we had above. So the example that we had above is kind of being shown here, where we have our BOH, HA right here, or AH, and we have ATP hydrolysis occurring right here. The terminal phosphate of ATP is being added on to B, and this is what you have here. And then AH comes in, and the phosphate is thrown off, being shown in this step right here. And you get A and B covalently attached to each other. So this is an example. This is kind of the example that we had above. Actually, it's the exact example. It just doesn't have all the delta G and all that other information in there. Now, if we look at an actual thing that happens in the cell, we have glutamic acid being converted to glutamine. It initially involves adding a phosphate to glutamic acid, and you get this high energy intermediate. Right, this is what it's called. And then you have the ammonia come in right here and replace the phosphate, and you get glutamine. So it's a conversion between two amino acids. If you're a little bit high on glutamic acid, a little bit low on glutamine, convert one amino acid to the other. The term that was being used above, activated carriers. What are these? So activated carriers are molecules. They're the ones where the reaction of hydrolyzing it, for example, or oxidizing it, will have a negative delta G standard or a negative delta G at cellular concentrations. So ATP is the ultimate activated carrier, because when you hydrolyze or you put it through a hydrolysis reaction and you get ADP plus PI, this is a delta G standard much less than 0. GTP is another activated carrier. NADH, NADPH, and FADH2, or these are also activated carriers. You get your activated carriers by having some process going in reverse of the process that releases energy. So obviously the reverse reaction, so let me just write out the reverse reaction of ADP plus PI going to ATP plus H2O, well, this is going to have a delta G standard much bigger than 0. This is not a favorable reaction at all. It's a very unfavorable reaction. So the process of making ATP from ADP is tied to breaking down carbohydrates in the foods we eat. And also GTP is made in that process also, and NADH is produced in that process also. The process of making these activated carriers eventually leads its way back to ATP and the products of cellular respiration, because that's ultimately the breaking down of food products, those big macromolecules that we eat, carbohydrates, proteins, amino acids, etc. As those enter cellular respiration, they're broken down, and the energy from breaking them down is used to make these activated carriers and activating the activated carriers. So in this case, taking ADP and adding a third phosphate and getting ATP, and then you can use ATP hydrolysis with this very negative delta G standard and use that and couple that to many reactions that are unfavorable and have them be pushed forward. So the energy to reconstitute the activated carrier pool is harvested from food through cellular respiration. And this is just a picture showing that. You have food molecules here, breaking them down, which oxidizing food molecules, breaking them down is energetically favorable. And you get your activated carrier and you basically activate it. In this case, if this was ATP, the activated carrier was ATP, you can think this is ADP, and now you added the third phosphate and you're at ATP. So then you can use that activated carrier and couple it to reactions that are unfavorable, right here, and push those reactions forward, because otherwise those reactions will not happen. But if you couple them, you add the two delta G's, and overall the delta G is negative, meaning that you are increasing entropy overall because the system's entropy is increasing, so the process can happen. If ATP hydrolysis to ADP is very favorable, then ADP plus PI to ATP plus H2 must be very unfavorable. Cellular respiration harvests the energy in the bonds of large molecules, such as glucose, to reconstitute the ATP pool from ADP. Okay, so this is very important. The statement that is being said here, reconstitute the ATP pool from ADP. So this pool of activated carriers, it's kept pretty constant. What do we mean by that? And this is what this statement is saying here. Let me just read it out. Note, ADP plus ADP plus AMP concentrations is a constant. Thus, as the concentration of one rises, the concentration of the others decrease. Therefore, high concentrations of ATP indicates the cell is in a high energy state, whereas high concentrations of ADP indicates the cell is in a low energy state. So let's ignore AMP for now and just stay with ADP and ATP, because you have this conversion of ATP plus H2O to ADP plus PI. And of course, this phosphate would be attached to another molecule. And then you have the ADP plus PI giving you ATP plus H2O. So these things are happening in the cell at the same time. ATP is being used, ATP is being made, right? So in a cell, the concentration of ATP and ADP, and also AMP, we're just going to leave AMP out of this for now. Because of these two reactions, if ATP concentrations go up, ADP concentrations go down. And the opposite scenario is if ATP concentrations go down, then ADP concentrations go up. So if ATP concentrations go down and ADP concentrations go up, well, the cell is at a low energy state because it doesn't have a lot of ATP, but it has a lot of ADP. Whereas if it has a lot of ATP and very low ADP, then the cell will be at a high energy state. Now, literally saying, just using one of them, saying that, oh, ADP concentrations are high, that means the cell is at a low energy state. Because if I know what the ADP concentrations are, I can know what the ATP concentrations are because they're connected to each other, and vice versa. So cellular respiration in eukaryotic cells. So we're going to cover glycolysis and the citric acid cycle to some extent in this section, in section 1, and then we're going to cover oxidative phosphorylation in more detail in section 2. But we're just going to touch upon oxidative phosphorylation in this section, just a quick touch up on it. So if I was going to take sugar and simply burn it, full oxidation of sugar to CO2 and H2O, meaning that if I had a 6-carbon sugar, I get 6 CO2s, all that energy from combustion will be lost as heat. And that's what's being shown here. Right? And you can burn sugar. Just put a little bit of sugar, although I would not recommend you try this at home unless you have an outdoor kitchen and you're being super safe. Just don't try it. Anyway, sugar can be burned. Also, there's a large energy of activation barrier to overcome, and that can be problematic, especially in the cell. So what cellular respiration does, it does the exact same thing as full oxidation of sugars and other molecules to CO2 and H2O, like combustion would do, but it does it in a stepwise manner. So you're starting basically at the same place, you're ending up in the same place, but you're doing it in a very stepwise manner where you have smaller energies of activation, EAs, and you get to harvest some of the energy that is being released by each of these reactions. Cellular respiration has 3 distinct stages. So let's look at stage 1. So stage 1 is glycolysis. In glycolysis, glucose, which is 6 carbons. So if you're going to start at the top of glycolysis, at the top of cellular respiration, it starts off with glucose. Okay, so we're just going to start at the very top with glucose. Glucose, which is 6 carbons, is broken down to 2 molecules of pyruvate. So for glycolysis, glucose enters 6 carbons, and you get 2 molecules of pyruvate. Now that's still 6 carbons. You just have 2 molecules of pyruvate, which is 2 times 3 carbons, 6 carbons. So you haven't lost any carbons here. No CO2 is being produced. This process uses 2 ATP molecules and produces 4 for a net of 2 ATP. So 2 ATP molecules are used, 4 are produced. 4 minus 2 gives you 2. So overall 2 ATP molecules are produced from this process. Also 2 NADH molecules are produced. Remember, NADH is an activated carrier. So here we have a little outline, basically a quick synopsis of glycolysis. And we're going to look at glycolysis in a little bit more detail later, but let's take a quick look. Glucose enters. You have one step, step 1, where ATP is attached to it, or glucose is phosphorylated. And then you have step 2, step 3, another ATP is utilized, and you get this molecule, fructose 1,6-bisphosphate. Now, why is this not called diphosphate? It's called bisphosphate because the phosphate groups are on 2 separate parts of the molecule. They're not attached to each other in series. If it was something like this, right, and the rest of the molecule are, then this would be a diphosphate. Now, at this point, fructose 1,6-bisphosphate is broken down to 2 molecules of glyceraldehyde 3-phosphate. And glyceraldehyde 3-phosphate is typically called GAP. So you get 2 GAP molecules, then these 2 GAP molecules start to produce ATP. You have step 6, 7, 8, 9, 10. You get NADH, 1 NADH per GAP molecule, and 2 ATP. But since you are doing this twice, you get 2 NADH molecules and 4 ATP. And that leads to a net of 2 ATP overall. So there's a bit of a energy investment steps here where ATP is used. You have the cleavage, and then you have the energy generation steps. Alright guys, we'll stop here and continue with the citric acid cycle later, okay? Take care guys, bye bye.